started. This is the second part of this international forum. En esta sesión,
So during this session, this is a pretty interesting session. We will unpack a few interesting concepts about diagnosis, diagnostic methods or diagnosing methods that developing nations face in order to identify these emerging issues. And in order to do that, we have with us a person with a lot of experience, with a high sense of care, which is Dr. Ian Lipk, a great person, investigators. He's a John Snow professor in epidemiology. He is from the Columbia University Public Health Care University, a very important university in the United States, in New York. He is also the director of GAP, which is a global alliance for the prevention of pandemics, with international recognition and a vision of joining efforts for several nations, especially in Africa and Asia. This is a person who has a lot of experience. He's running a lab that is pretty advanced. This lab does pathogen research, new pathogen research. He's developed platforms for viral pathogens such as bacteria and some parasites. Technology that allows us febrile, to even with febrile entities without a diagnosis and see the causes easily. He's been a pioneer in several areas. With MERS, for example, he has a lot of stories, amazing stories, stories about camels and with Arabic food. He is definitely a great friend. And to me, it is an honor to introduce Dr. Ian Lipk. I could spend 2 days giving you the list of all the work he's done in his publications. So without further ado, I will introduce Dr. Lipk. I will ask him to please come on up and give us a review of this great adventure, which is identifying new pathogens.
Thank you very much, Guillermo. You also have wonderful stories. I'm embarrassed to be speaking in English. I understand more Spanish than I can say, so be careful. So this first slide illustrates the fact that although we mostly talk about viruses, there are many different pathogens that are important. And if you think in terms of yourselves as hunters, You put them up next to your fireplace, like deer heads and things like this. And the whole field of infectious disease epidemiology really began in the 1850s with John Snow and the implication of the Broad Street pump. And you can go to Broad Street in London today and you will see a pump. It is not the actual pump. The actual pump is inside the London School of Hygiene and Tropical Medicine. It's also not true that John Snow went and broke the pump handle off to stop the epidemic, but it is a great cartoon and it's something that we all aspire to. Now, you've seen various versions of this map. I update it periodically, as you all do, and the latest editions are monkeypox and polio, and I haven't really put down a place for them yet because I'm not exactly sure where they originated, but they're clearly now global. And the tools that we primarily use now are sequencers, and I will be talking with you about a variety of ways in which you can use sequencing more cheaply, more rapidly, and so forth. Now, my whole introduction to this began when I was a neurology resident. I did an internal medicine residency. I finished it, and I wanted to do what Paul Farmer did 20 years later, but it wasn't possible. They said I didn't have any relevant experience. So then I went and I did neurology at UCSF, and I was the only neurologist who was willing to see these patients because everybody was afraid they were going to get infected. And what struck me most was the fact that it took 2 years to figure out what it was. So I then went to work with Michael Oldstone and Floyd Bloom, and there was a virus that they thought was a virus that was implicated in manic depression or bipolar disorder, but nobody knew what it was. So I said, we can solve this. It's not that big a deal, right? With the sort of naivete of a clinician. And so I undertook subtractive cloning, which took me many, many years. And ultimately, we identified these clones. We didn't sequence them because we didn't have access to sequencing. But it took us 5 years, us being me, myself, and Ian, but it took a very, very long time. When you contrast this with the introduction of high throughput sequencing with 454, in the early 2000s, this took 2 weeks to identify this particular arenavirus that was found by Gustavo Palacios, who is incidentally an Argentinian. So, and when we first put this out, people said, but you haven't proven that it causes disease. And now people publish sequences all the time without having to go through all of the things that we were required to do. And then Lujo came about, and Lujo took us about a week. It was still 454. Many of you may remember this. It was probably the most lethal infection ever discovered because there was a single woman who became infected. She infected the paramedic who lifted her from Zambia to Johannesburg. The nurse on the other end, the person who cleaned the room, all 4 of those people died. And the last individual, a 47-year-old nurse who took care of the people who became infected in Johannesburg, received ribavirin, and she's the only one who survived. And to my knowledge still, this is the only example where we've actually, in the middle of an outbreak, stopped it in its tracks. That doesn't mean we shouldn't be doing this, but I'm just saying getting from that to a solution is difficult. So there's Roche 454 and Illumina and Ion Torrent, which is basically a big pH meter, and the Nanopore MinION. And then, of course, we have the tricorder, which we're all excited to see. But just to illustrate the point that all of this is going to continue to improve and change. One of the questions that frequently comes up is, how much viral diversity is there? We tried to estimate this a few years ago, and this was based on consensus PCR. So it didn't even use high throughput sequencing, but we figured there are at least 300,000 viruses yet to be found. In mammals, it's somewhere between that and 1 million, and that the cost for this would be about $6.3 billion to find them all. And I actually had some calls from various people who said, how do I invest in this? Now, this was the basis for the Global Virome Project, which we've since sort of abandoned because it's very difficult to get from finding a sequence to implicating something in disease. And this program is an example of what will get us there. And that's why we're working here. Now, we've found over 2,000 viruses so far. I would say 1% of them are either clinically or economically important. And if you combine this with all the other laboratories around the world, we're talking about hundreds of thousands of viruses. So the question is, how do you prioritize? Where do you invest? Because there isn't any good way that I know of yet to say that this particular finding we have is going to be important, except through surveillance. So we've been trying to find ways to make surveillance faster and cheaper and more democratic. This is a system we developed about, it's 8-9 years ago now, which is capture sequencing. It's basically like exon capture. And we've taught, probably 100 people how to use it now. It's very, very easy. You simply have probes that occupy the entire viral sequence space. You bring them down with magnetic beads. And this has two advantages. One, the sensitivity is equivalent to real-time PCR. I know that's hard to believe, but it's true. And I'll show you that in a moment. But in addition, because all you're bringing down is relevant sequence, you don't waste a lot of your sequencing power on irrelevant human sequence. And in addition, because you're not bringing down human sequence, you don't have problems with invading people's privacy. So it's much easier to get people to agree to participate. And this is what it looks like, a cartoon that shows it's really very simple, but you can see you have a 2 to 3 order of magnitude enrichment using this approach with a wide range of viruses, single-stranded, double-stranded, DNA, RNA, it works. You can do the same thing for bacteria, and I'll give you some practical examples. Here you get about a 2 order of magnitude to 2.5 order of magnitude enrichment for relevant sequences, and you can target this for antimicrobial resistance or anything you wish. So again, it just gets you where you want to go faster. And here's the example of what I mean with that. So just taking a mini-seq as an example, with a 4-hour run, 25 million reads of 150 nucleotides with 3 samples, if you take this through capture, you can get the whole thing done in 14 hours, which is amazing. And we can show you how to do this, and you can do it yourself. It's not that complicated. The bioinformatic analysis is also easier because you don't have a lot of irrelevant sequence from the environment, whether that's a human being or an animal or a fecal sample or whatever. So this greatly expands the number of labs that can do this kind of work. Now, we patented this not because we want to make a lot of money on it, but because if you don't patent it, somebody else can patent it, in which case you don't have access to your own technology. So anybody who wants to use this for public health, it is free. If you're a company, that's different, but it's now been through New York State Department of Health, and it's going forward for FDA licensure as well. The other thing that's very important for making links between infection and disease is antibodies. And we all know now about that because everybody tracks that, and everybody in this room has done this kind of work, but there hasn't been a way until recently to examine a wide multiplex of different possibilities. We began working with a company called Nimblegen, which became Nimble Therapeutics, which then became part of Roche, and now they don't even make these arrays anymore. So all I have is what is stocked up. So now we've had to shift to a phage array system, which is not as good, and I'll show you why in a moment. But this allows you to look at hundreds of thousands of potential exposures. I'll give you a few examples, of which there are now a dozen. This was an effort to develop an ELISA for detection of Zika in women who'd been exposed in Colombia. We did this in support of an NIH program, and it is extraordinarily powerful, as you can see, and very descriptive because you can differentiate different flavivirus infections. You don't have problems with cross-reactivity. It's as good, for example, as neutralization assays. With acute flaccid myelitis, which is a paralytic disease of kids, which has a sort of a biphasic incidence, there were 2 groups simultaneously working on this. One was using phage display, one was using arrays. And the phage display approach got you to enterovirus. But this took us to enterovirus D68. And that was because these peptides are displayed across the entire proteome of the virus. And you can look at overlapping fragments so that when you get signal, it's very hot and you could see this is the peptide that's important. And this then has been able to guide a lot of work that many people are now doing with immunotherapeutics. This is the phage display system. This is actually a very old system. It was first really developed in the 60s and the 70s, but it's been modified now for the purposes of doing human serology, whether you're looking for autoantibodies or you're looking for infectious agent exposures. Because we no longer can get these arrays readily, we're trying to take phage display and make it more granular. And this is a comparison in patients with myalgic encephalomyelitis chronic fatigue, which is a disease that some people looks like PASC or long COVID, if you prefer. And what we've tried to do here is to look at reactivation. And because these are very discrete, we can say not only that the titer goes up in the case versus the control, but you can look at immunoactivity directed against specific genetic markers for reactivation, immediate early genes, for example. And we've done this with convalescent COVID patients. Now it's not perfect overlap because you can see the peptide arrays are indicated in that greenish color, and the browner one is the phage display. The peptide array is still superior, but as I say, unless somebody decides to start making those again, it becomes difficult. I'm going to give you an example how we put all of these together to investigate encephalitis in Uttar Pradesh in northern India. So there were these regular outbreaks of encephalitis. You can see here there are 2 kids to a bed. The families are waiting there in the waiting room. Sumi Swaminathan, who's now the scientific director of the WHO, who was then the head of ICMR, asked us to go in there and participate and try to sort it out. They were building a hospital just for these kids because there were so many cases. But in this case, you can see, you know, this was useful for other purposes. It's now become the COVID hospital. So the reason this area is so heavily afflicted is that there's regular flooding, as you can see here now, right up toward the border with Nepal. And these are children who are paid a few rupees a day to collect and kill rodents. So they get infected by the lice and the phlebotomist flies on these rodents. And these children, the Musahar, are the lowest socioeconomic class. They're not even in the caste system. They're typically tribal. Now, when we went and looked at the spinal fluid of these children, they had already been studied by US CDC and others, and nobody had found anything in this group. There was the unexplained group. And you can see we found things that you should have seen, like herpes simplex and parvoviruses and hepatitis and mumps and so forth. But what was most striking to us is that we found sequences for 2 different types of rickettsia. And when we combine that with the peptide arrays, using antibody-based and molecular methods, we're able to resolve 50% of those unexplained cases and implicate rickettsial species. This work is not published, but it's being used now to direct early antibiotic therapy of these kids. Here's another example, but here I'm emphasizing something slightly different, and that is reagent contamination. When you begin doing these high throughput sequencing methods, you find a lot of things that are present in all the reagents that you use. So we came up with a way to clean it up. So we radiate plastics and buffers. We digest anything that can't be radiated with 4 base cutters. And then that leaves us with little fragments that don't enter the sequencing. And that then allowed us to implicate this one particular bacterium, Panibacillus, in hydrocephalus. So then you could go back, create a PCR assay, and find, in fact, evidence that this is the case. So again, this is an antibiotic treatable illness that otherwise results in this horrific situation. I'm not going to talk about this because I think this is for me to all of you, but I will focus a little bit on climate change. We've been seeing in the United States a dramatic increase in tick-borne diseases, and I'm confident that you will too. And the reason for this is that the growing period of ticks is becoming extended and their geographic range is increasing too. Travel. You saw other earlier presentations talked about travel. This is just coming into the greater New York area. But if we focus on the US-Mexican border, it's very clear that whatever is in Mexico is in the US, and whatever is in the US is in Mexico. So we have to work together because of all these different crossings where you can see examples where there are opportunities for exchange of infectious agents. Now, Tony Fauci and Cliff talked a little bit about wildlife trade. It's important to realize that there's a legal wildlife trade and there's an illegal wildlife trade. And it's all over the world. It's not restricted to China. It's not restricted to Mexico. And when we've looked at other parts of the world and we've looked at ourselves, we found examples of this. So these are what was circulating in the Wuhan market prior to the beginning of the pandemic. But in JFK, when we went looking at animal parts that were being imported into Queens, we found primate heads and all kinds of things. And these were infected with a wide range of bacteria and viruses. The other thing that we don't focus on quite as much as we should is all the various rodents that are existing inside of our major urban centers. So these are rats collected in Manhattan between Chinatown and City Hall. And when we looked at these animals, we found that they had a wide range of bacteria. And many of these bacteria were antibiotic resistant. More important still, because the rats tend to stay outside, when we looked at mice, because mice actually get inside your house, and you can see them here inside somebody's kitchen, we found that these animals had, and this is in every borough in Manhattan, we found that they had lots of bacterial infections with antimicrobial resistance. I'm not going to talk about COVID except to say that the thing that people are not focusing on enough, to my mind, is these post-COVID sequelae. And this, I can tell you, is one of the first people in the US to encounter this virus, makes you, can make you very, very sick and make it very, very difficult for you ever to fully recover. Cognitive dysfunction, all sorts of other challenges. And we don't fully understand it, but it's beginning to look like it's a combination of structural damage, which we think we understand, and then various metabolic disturbances and induction of a lot of immunity, which is going to be, I think, easier to treat. We're working on it. Now, the program that I'm representing is called GAP, the Global Alliance for Preventing Pandemics. This was started by Jeff Scholl's investment. Jeff was also the premier, the first investor in the movie Contagion, that I'm sure some of you have seen. And the idea is to help various other organizations build global immune systems with a sort of a democratic approach whereby people are empowered to do their own sorts of work wherever they happen to be. And we're now working in many different places, and people have been successful, and NIH has been a great partner, and Guillermo's been a great partner, and we have all sorts of very interesting programs. And you can see there is Mongolia. We're not working with them quite so much anymore. I guess they're now working primarily with NIH. And this is the way it works. People come in, they work in the lab, they learn how to use these various tools, and then they go home. And then they write various articles, and we try to emphasize senior authorships and primary authorships from the individuals who do that work. Now, the one thing that I do want to close in is talking a little bit about the dark side of all this pathogen discovery. When you start using these tools, you're going to find things that aren't really true. This happened with measles, with MMR vaccine and autism back in the late 1990s, and it has taken decades, and we're still confronting this as a problem. It happened with chronic fatigue syndrome. It's happening with a wide range of other disorders as well. And social media has amplified a lot of this. We talked a little bit about this in Contagion. Some of you will remember Jude Law, who played a very interesting character in that particular film. Now, I think scientists don't spend enough time on social media, or the wrong scientists, I should say, spend time on social media. But we need to find ways in which we can actually promote what we think would be appropriate responses. Early during the course of the pandemic in March of 2020, and I don't have the ability to control this, I don't think. Can you start that first film on the left? So we got the cast of Contagion to try to talk about ways in which we could promote health. Thank you. And they were all eager to do it. So this was Matt Damon, and then we had, and obviously Larry Fishburne and others talking about various ways in which you could prevent contagion. And Kate Winslet stopped there and start the next one. When we started talking about vaccines, because I knew that vaccines were going to be a problem, I went back to the cast and they said to me, look, why was anybody going to listen to me? I'm just an actor. Get people who look like members of their own community. So we found Black and brown people and Jewish people and priests and others who had the confidence of their communities to promote this. I think you can shut it off now. Thank you. But this is our biggest problem, I think, is trying to convey that people need to do the right thing, whatever that is. I don't have control. Something I'm actually used to, but in this case, it's important. Can you advance this, please? Thank you. It's interesting to see what they look like without makeup, isn't it? It gives us all hope. Yeah, exactly. Okay, so now I want to mention one other thing. This is sort of the future of virology. The Department of Defense had this meeting in March of '99, just prior to the onset of West Nile virus. And they had one animal virologist, myself, a plant virologist. Nobody talks very much about threats to food security. And we found, for example, viruses that kill salmon and tilapia and so forth. And they also had somebody talking about computer viruses, because there was recognition that there were similarities in all of these. And this just gives you an idea of how things spread around. So TikTok, tweets, Facebook, absolutely extraordinary, far more powerful than conventional media. Next slide, please. So we try to do what we do in terms of education through print, but next slide, please. And this is the final slide. I think it's much more powerful to use media. So No Time to Die had a biological weapon, Utopia, Contagion, and Plague Inc., which was a game. And it's a very easy way to expose people to some of the concepts that we want to share. Thank you very much for your attention.